This is Genius Pro Online School, and this is Tutor PVS. Here in this section, join me in biology as we talk about enzyme. It will be the easiest video which you are going to find to explain enzymes. All right. What do we need to know about enzymes first of all? All right. Number one, we need to know that enzymes, they control the rate of reactions. All right. They do what? They control. Enzymes control the rate of reaction. What else? They are biological catalysts. They are biological catalysts. What else? They speed up. They speed up reactions. In the living organisms. That is what enzymes are. So how can you come up with a complete definition of the word enzyme? Well, here's it. We can combine this. We can say enzymes are biological catalysts that control the rate of reaction and speed up the rate of reaction in living organisms. Or you can say these are biological catalysts that control the rate and also speed up, all right, the reaction rate in the living organisms. These three things should be there as you are defining enzymes. All right, now enzyme action. How do enzymes work? So let's say we have an enzyme here. Okay, so this is the enzyme. Now, an enzyme will always act on a substrate. All right, the enzyme will always act on the substrate. So let's say our substrate is this. Okay, this is our substrate. So when this substrate comes, it has to fit exactly on this part there. All right, so this part here is what we're calling the active site, and the rest is what we call the allosteric site, the rest of this part of the enzyme. So this one is a substrate. All right, so these two, when they come together or when the enzyme acts on it, all right, you form something complete. All right, so you end up forming something like this. But take note that somewhere here we had a substrate. All right, somewhere here we had what? A substrate. That's like a substrate there. And I will represent the enzyme just like the way it was. This structure here is what we call the enzyme substrate complex. All right, now this can still go on and go on because we need products. So it's got that the enzyme will remain the way it is. The enzyme will remain the way it is. Okay, then there will be plus. Let me just write them here. We are going to have products. So this thing of ours here, it will be broken down into two. So we can have one of that nature and the other one of that nature. All right, so those will be broken down. Okay, these, you can say the enzyme has then changed the shape. So these are what we are calling products. This is still the enzyme. Hence, enzymes do not change at the end of the reaction. That means to this definition we add they are unchanged at the end of the reaction. We are now coming up with a complete definition of an enzyme. So for now, the four parts now make up the definition of an enzyme. Okay. All right. Now, what else do you have to know? In this case, enzymes are specific. When we say enzymes are specific, we mean they can only catalyze one reaction. So enzymes specificity. Now, this one can be explained properly using the lock and key. So we're going to use the lock and key hypothesis or mechanism. Others they call it mechanism. So how do you use the lock and key hypothesis to explain the enzyme specificity? Well, this hypothesis states that the enzyme, the substrate, it fits exactly on the active site of the enzyme. Like we saw, the substrate will fit exactly on the active site of the enzyme, just like the way a key fits into a lock. Why? Because the key and the lock are complementary. Are we there? That's what the lock and key hypothesis is about. All right. So in our case, all right, whichever acts on another thing is the key. So the key, it is what acts, all right, on the lock. So our substrate becomes the key, and the active site of the, not the entire, the active site of the enzyme becomes the lock. Others, they just say this, the enzyme is the lock. Well, yeah, but to be specific, it's just the active site, which is the lock. All right, so you've known which one is a lock, you've known which one is an enzyme. Perfect, right? You can explain what enzyme specificity is, because it just means the substrate will fit on the active site of the enzyme, just like the way a lock fits into what, so the key fits into the lock, because the two are complementary. All right. Now, there are two factors that I want to talk about in terms of enzymes that affect the enzyme activity. That is temperature and pH. These are some of the crucial factors that affect enzyme what? Activity. So you can, if you are copying, you can just write factors that affect the enzyme activity. All right. Number, temperature. Now, anytime you hear temperatures, think of this. Talk about kinetic energy. Talk about kinetic what? Energy. Talk about collision of the particles. As you are explaining how temperature affects, talk about both when temperature is low and when temperature is high. Don't just talk about one side. No. Are we there, right? Then talk about what happens when it is low and when it is high. Then you have given the complete description of how temperature affects the enzyme activity. You do the same for pH. Now, pH, you don't talk about kinetic energy. You talk about the regions. The regions, all right? Okay, so here you talk about, let me be specific, you talk about acidity. You talk about acidic and alkaline regions. Sorry. For example, there are some regions that are alkaline in nature, hence enzymes that work best in the alkaline medium, they're going to work well there. There are enzymes that work best in the acidic medium, and hence they can't work well in the what? Alkaline what? Region. You need to draw that distinction. Okay? At temperature, you need to know that they work best. They work best at 40 degrees Celsius, and others, I think, let me just say from the range of 36 to 40, others, they work well in that range degrees Celsius. All right, that's just like the optimal temperature. All right, they are specific. Those are also characteristics. They speed up the reaction. They work best at 40. They are specific. They are made up of proteins, like we said, they are protein in nature. They are biological catalysts. They are not changed at the end of the reaction. They are quite a number. All right. Now, enzymes can also be, well, used in real life. All right. We can use them in washing powders. We can use them in production of fructose. We can also use it in making leather shoes. Can also be used in tenderizing of meat. There are quite a number. Enzymes can also be used in baking and brewing. For example, the zymes, the one which is found in yeast, you can use it in baking and brewing. So there are quite a number of uses for enzymes. You can look them up there. But I think this has summarized everything, and I hope I'll see you in another one. This is just, the summary of everything. That's what we do.